we'll start. So I'd like to open this, it's at Hupo Education and Training Auditorium. And I'd like to open this session with an introduction for Sue Weintraub, who will be our speaker this month. So she's a professor in the Department of Biochemistry and Structural Biology at the University of Texas Health Sciences Center at San Antonio. To this audience, though, she is probably best known for her past work as president of ASMS, vice president of HUPO, and all of the service she's performed for the community. But in addition, over the last 40 years, she's chaired over 30 NIH study sections. That's kind of mind-boggling to me, not just participated on, but chaired 30 study sections. And then she's also been awarded 9 S10. So these are very large instrument grants. So today she's actually going to talk to us about some of the best practices in preparing grants and submitting grants. So I'm really excited about this session. I'm looking forward to getting all questions afterwards too. All right, Sue, give it a go.
Okay, thank you so much, Brian. I'm delighted to be here to be able to talk to you about grant application preparation. It's obviously the key to funding your research. And there are many aspects that once you have developed good practices, you really can be very successful. I wanted to show you a photo here of my very first solo grant application. This was in 1982. I'd like to say I was 5 then, but I think that doesn't work. This was for a double focusing, high resolution, reverse geometry mass spectrometer. You can see the cost is $250,000. This was prepared on an IBM Selectric typewriter, and if you had typos, you had to correct using a liquid called whiteout. And you can see I've written here, this has the original typing and original letters. This is still in my drawer. And this is blanked out because we used to put our social security numbers on there. That was required. And that really was very exciting for me because, as you'll notice here, I was a research assistant professor. And this was my first funding, and it let my lab really get started. And that was 1982, the first year of the S10 program. And I hope to tell you a little bit about S10 at the end. Now, I always like to start with something a little light to introduce, and I thought these kid scientists were cute and preparing a gravitational force experiment. And of course, natural to them should be, do you think we can submit a grant application for this study? And it looks like a very simple study. Yes, you have a feather, light as a feather, and a tennis ball, and how does gravity affect that? But if you really look closely, there's other factors that need to be considered. For example, and I hope you can see my cursor, but this window is open. And so you have the airflow from the window is probably influencing the movement of the feather, probably not so much of the ball. And clearly there's other factors that the applicants would have to take into consideration, but probably the reviewers would be astute enough to know. So the key is going to be, how do you prepare a proposal to get your experiments done and be convincing to your reviewers? I'm mainly going to be talking about NIH grant applications, but most of the elements are really pertinent to most other funding agencies, maybe not the specifics. But since many of you will be applying to NIH, I thought it important for you to see what the general steps are, because a lot of people don't think about, well, what happens from the beginning to the end. And what you see is you're preparing your proposal, and if you're at a institution with good support, they're going to be helping you with the practical aspects. It will be submitted electronically these days to NIH, not boxes of those paper copies. And I got to tell you how many times we raced. It had to be postmarked by a certain time, date, and you would race to the airport facility, the post office, so that you could get it before midnight. Then there's actual assessment at NIH to make sure that the document can move forward in the process. And this is things like, have you included all of the necessary elements? But even if you've gone way over on page numbers and not followed the instructions, it may be returned and not reviewed at all. Then, based on the topic, it will be assigned to a review panel, which we tend to call a study section. The study section is headed by a scientific review officer, an SRO, who will then assign reviewers. Normally, it's 3 reviewers. One often is designated as primary. The reviewers will then prepare their critiques using certain forms, and often in bullet point format. And then they will upload those for viewing by the rest of the panel members prior to the meeting. I will say, most times you're so busy, you don't really have time to look at the other critiques at that time. Then you meet as a study section, and normally about 50% of the proposals are discussed and scored. And this is based on preliminary scores that are given by the assigned reviewers. I'll talk about that a little bit more later. Then there's an actual study section meeting. Used to be almost entirely in person. They were, for many reasons, of course, during COVID, all virtual, and some are moving back to in person. And I'll talk about the process there. But then after their scores, they're released to the applicants online. Don't have to wait for the mail like we used to, pretty quickly. And then a summary statement with all the critiques of the reviewers is prepared by the SRO and then released online to the applicants. There is a brief window after this where if you see an actual factual error in the critique, you can appeal. Then all of this goes to what's called a council for the Institute of the funding agency. And then eventually the decision is made for funding by the program officer. Now, I highlight here program officer because you see the program officer at the end, but they could also have a very important role at the early stages. How do you start? Well, don't put down a single word yet, not even the title. Think about the concept, and this is absolutely key. And regardless of how many proposals you've prepared, you need to allow sufficient time to think and plan and work out what you're going to do. Decide on the main focus of the proposal, just in general, broad strokes. Of typical examples, it could be an extension of your current or prior research. Important, especially for young investigators, to remember, if you're following up on your graduate or postdoctoral research, you really need to get permission from your previous mentor. Whether this has to be in writing or not, it's not for sure, but you have to be sure it's okay for you to be working on it. You might have a completely new idea you want to follow up on, or you may look at the funding opportunities and adjust your ideas to fit something that there is a funding opportunity for. Right. A lot of people don't think about this. You better conduct a thorough literature search, especially if it's a new idea. Make sure someone hasn't already done it. And then you want to formulate a hypothesis, because many grant applications do best if they are hypothesis driven. We really can't have what they call fishing expeditions. Now, fortunately, discovery proteomics is more accepted and no longer called fishing, but that's not enough for an entire grant application. And then you need to decide on a set of specific aims. That's what they're called in an NIH proposal and in some other types of funding. But in general, there's always going to be some specified aims of what you want to do. Now, some people like to make this final right at the beginning, but I strongly recommend that you don't finalize the wording until your plans for the proposal are absolutely decided. Because I see a large number of grant applications as a director of an institutional core where we're going to be providing help and support. And many times I'll find some issues with the specific aims, either in the wording, it's not clear, or the methodology. And if that has already been finalized elsewhere in the proposal, you have to make sure that everything is consistent. So you want to have it set what you plan to do, but not finalize it. All right, funding source. Where are you going to submit this? It's not all just NIH. There's many different funding agencies that may be very appropriate for your specific topic. And you may not know of many of them. So there's funding agency websites, and really excellent way here at our institution and elsewhere is you can subscribe to funding opportunity updates. Our grants management department's sponsored programs sends out several times a week list of new opportunities, but you can also go to the different funding agencies and sign up directly. Now, here's something that many people don't think about, but this is not a completely anonymous process from the funding agency and the applicant. The reviewers are. And what is really a great idea to do is once you've picked a target agency, you can find out contact information and contact them. And in NIH, this is called a program officer. And that's why I said that this person is also important at the beginning, because you can send them by email. They're very busy. You don't want to just call them. And prepare a one-page description of your project. If you have some preliminary specific aims, outline of the experimental design. And now they're not going to tell you, oh, yes, this is fundable. But what they will tell you is if it does or does not fit with the interests of that particular funding opportunity or agency. And many times, if it doesn't, they will be able to guide you to an alternative. So it's really important to make this contact early. And what is their goal? Their goal is to fund outstanding research. So it's a great connection to make. Here's a list of the main NIH research program types. And I've highlighted the 3 that would be of interest, presumably to this group. The one we hear about most is called R01. It's initiated by an investigator, and it's a full research project, generally covering 3, 4, or 5 years, depending on the project and the funding opportunity. And then there's a exploratory called R21 that's also investigator initiated. And even though one might think that this is developmental and probably easier to get, it is usually harder to get funding through the R21 mechanism. There are often benefits for being a first-time and young investigator in a lot of these programs. And then here is the S10, which is support for shared instrumentation. Now, through this, I'm going to mention a little bit about a colleague, collaborator, friend, Julie Thomas, who's at Rochester Institute of Technology. She's an associate professor there. But I first met her when she was a postdoc in San Antonio. And Julie works on bacteriophage. Bacteriophage are really fascinating organisms because of their diversity and other aspects, their interest in biology, but also the fact that bacteriophage are viruses that live on bacteria. And they can be used to treat antibiotic-resistant bacterial infections. But there's a whole lot to their study, and that's what Julie has been doing. And I will tell you a little bit about an R15 proposal she had that got funded. And an R15 is essentially styled like an R01, but it has an added academic and training component to it. Now, we're still at the preparation step. I haven't really written anything down, maybe some notes. Think about your experimental design for the concept you've decided on. What techniques and resources are you going to need? What do you have on hand and what needs to be acquired or learned? And how feasible are the techniques? This comes to mind recently. A clinician scientist at a nearby hospital was partnering with some others, and she wanted to submit a grant application to the Department of Defense to study something related to battlefield injury. And she was referred to me because I run a mass spectrometry core. And in talking to her, she described her project. It sounded very reasonable. And she says, okay, well, I want to do metabolomics and proteomics. I said, can we be a little more specific? Oh, no, I want to look at blood, and I want to get a very comprehensive list of all the proteins and see how they're changing across individuals. And the same thing for all the metabolites. And, I sat there, I thought, how can I tell her? This is absolutely not possible. So we spent a long time working through, and in the end, she's going to propose to look at proteomics of PBMCs, and some targeted metabolomic small molecule analysis. But, you know, beforehand, if she had submitted that, that's dead in the water. All right, is the project innovative? Yes, it needs to be innovative to be interesting, but not beyond belief, not pie in the sky that is just so impossible to imagine it working. Do you have the expertise for this study? For all of it, for part of it? If not, how will you get the necessary skills? You can't be expected to know everything, but you need to have the necessary basis. And do you have the instrumentation, equipment, or infrastructure? If not, how will you get it? Some grants, applications, and funding opportunities have the option of getting some small equipment, but certainly not something very large. And these lead into the question of, do you need a co-investigator, collaborator, or consultant? Now for Julie, this was very clear. Julie is an expert in isolating, propagating, working with bacteriophage, and amazingly good at the informatics associated with the project. But the first thing she needs is genome sequencing. So she needs a core to help her with that, or a service. She needs mass spectrometry. Well, fortunately, we've been working together, so I can provide that. And then for structure, she needs cryo-electron microscopy. And that she also has a collaborator to work on. But there's a really key important point here. It is still Julie's project because she needs to be developing the experiments, the experimental design, taking the results, and coming up with biological conclusions. She's not serving as a concierge and just sending out samples and receiving results. And that's an important point that needs to be made clear, that the PI is actually leading the project. Then I asked, what's the impact on the budget? Well, this is because it depends on the amount of effort of this co-investigator, collaborator, or consultant, and they're obviously at different levels. If, for example, my core is just going to analyze a couple proteins for identity, I don't need to be anything more than a consultant. But if I'm a co-investigator or a major collaborator, they're going to want to see what's called percent effort. And percent effort is associated with some budget. So the amount of percent effort has to be commensurate with the person's contribution to the project, and that can impact your budget. So it all has to go together. Another point, which may seem trivial, but you need an NIH biosketch for each of the people who are listed in this key personnel section, and letters of support that document that they're willing to participate. And it also provides details about their contribution. So for example, on my letters of support, I tell a whole lot about the core and the technology and the methodology we use so that the investigator doesn't have to put it and use up space. Well, a practical consideration is your collaborator, co-investigator going to be available at the time of submission? You can't do an update. So if you don't have any information from that person, they need to be able to get that to you in a timely way. Preliminary data, that's another tricky point. I put, is it needed, allowed for the target program? Well, it's almost always needed, but in a few very, very rare grant applications, it's not allowed. So it's something you just need to verify, but you have to decide, how much do you put in? Some people have the project almost completed before they're putting in the funding, which is ridiculous, but you don't want to put too much. But what this does is demonstrate your technical capabilities, and it documents project feasibility, at least for the beginning. How much is enough? There's no easy answer there. All right, I say this, you're not alone. Accept help from others. This is not like a student project where you have to do the whole thing yourself. Be sure you talk to colleagues who have previously applied, especially to the agency that you're targeting, and especially those who have been successful. Now, if you're at a good-sized institution, you're likely to have a full office of sponsored programs, grants management, and also they may even have programs where people can help evaluate your proposal, even at the very early specific aim stage. We have something called Grant Seekers that's really fantastic. Now, it's important to keep in mind the NIH review criteria, because as you'll see, the elements of the proposal in a way parallel. And this is where I'll tell you a little bit about the review study section. The 3 assigned reviewers, after they have determined their critique and come up with the strengths and weaknesses, which is the way the critique is divided, there will be scores for the categories of significance, investigators, innovation, approach, and environment. NIH scoring, if you don't know, starts with 1 as the best and 10 as the worst. 1 meaning that there are essentially no flaws, everything is great. 2 is still a really excellent score. And if you get something 4, 5, and 6, that means you've got a lot of problems. At the beginning, these assigned reviewers will input their scores for these categories, and then they'll come up with an overall impact score that is not the average of the subcategories, but it has to be in line with them. So if you have all 1s here, you obviously can't put a 4 and vice versa. Now, these are preliminary scores that then will be seen by the other review panel. After the study section, there will be the assigned reviewer at the study section meeting. The assigned reviewer number 1 will present everything about your proposal, summarize and talk about strengths and weaknesses. And then reviewers 2 and 3 will talk about anything that they wish to add. There's discussion among the panel. And then the assigned reviewers will restate their overall impact score. It can be completely changed based on the discussion. And you can often get much more, the term is enthusiastic or less enthusiastic, depending on the other reviewers. Nobody says you all have to agree. You just have to make clear what your comments are. But then after that is done, all of the review panel members enter a score, and they do that privately. The score has to be within the range of the assigned reviewers, or you have to state that you are outside that range. And what is hoped that the assigned reviewers will adjust their critique to match their scores. And this is the overall impact score that you get when you hear about a score on a proposal. It is essentially the average of all of the assigned scores times 10. So if you got a score of 10, that meant everybody gave you a 1, which pretty unheard of, but on occasion. Some practical considerations as you're preparing to start writing. Remember, the big picture, the concept is important to sell, but you've got to really pay attention to the small details as well. And these are somewhat subtle in a way, but it has a major influence on the reviewer's opinion. The word is always their enthusiasm. And I will tell you that at a review panel, we always avoid what we jokingly call the F word, which is funding, because we're not considering funding a proposal, but its scientific merit. You really need to be consistent with style and formatting. Just think of something if you've got different fonts in different places, spacing, it doesn't look very neat. And it gives the indication that maybe you're not a careful researcher. Minimize the use of acronyms, because it's not a paper where you can go look back into abbreviations. Avoid the use of topic-related jargon. Remember, not all of the panel members are going to have in-depth knowledge of your field or technology. And what I also have here is be sure that your table and figure numbers are consistent with the text. It seems silly, but I can't tell you how many times I've seen someone took a table or a figure from a paper or from another grant, and they forgot to change the number relative to how they refer to it in the text. This is definitely not good. Be concise, but not at the expense of clarity. The key elements of the proposal, and I'll show you just a little bit about some, the abstract, which is obviously a summary, the specific aims, which we'll talk about, research strategy, you see this parallels the scoring, significance, innovation, and approach. Approach will have some background. It can have literature, review, and preliminary data of yours. And then the experimental design that's organized according to the specific aims. And it will also describe in detail. Now you have page limits, often 12. So you can't put every concentration of every solvent you're going to use, but you need to make it clear that you know what you're going to do. The methodology is well thought out. You know what the expected outcomes are. And you also need to list potential pitfalls and alternatives. Now, this is another tricky place, because if there's an obvious pitfall, like you're trying to isolate a protein and maybe you can't get it active, you need to have an alternative. But you don't want to think up some obscure pitfall that the reviewer isn't going to think of and get them going off in the wrong and negative direction. But it's important to be sure that any realistic pitfall is mentioned, and a timeline. How does it fit together? Abstract to me is absolutely essential. Some people just don't pay much attention when they're preparing their proposal and just throw together a brief summary. There's only space for about 450 words, but to me, this is something that's read by many of the reviewers, because what happens at the panel is you really haven't had time to look at the other proposals. Reviewers 1, 2, and 3 will be talking about it, but you have access as a reviewer to all of the electronic versions. So you tend to pull it up and start scanning it. And what's the first thing to see is the abstract. Now, when we had paper copies, it absolutely was the first, because that was right after the cover page. But you need to summarize the key components of the project and excite the reviewers about the project. Why does this have to be done? Why is it essential? How will this move forward our knowledge of biomedicine or of biology? And then the specific aims is 1 page for NIH proposals. You start with a brief summary of the project, and you highlight any of your noteworthy previous findings. Now, this is just 1 page, so you don't have a lot of space. Then you state your overarching hypothesis, and I emphasize again, hypothesis. If you can't put your concept into the form of a hypothesis, you have a much less chance of being successful. You have to be asking a question in that term. Then you list the individual aims. Usually it's not more than 3. If you have many more than 3, they may say that the project is too diffuse or it's overambitious. And if you have too few, you may not really be doing enough. And within each aim, you have a very brief sentence about the strategy or methodology that you're going to use, and then expected outcomes, all in 1 page. Now for Julie's, the R15 I was telling you about, she started off by saying and posing these questions. These are not her aims directly, but what are the locales of the many different proteins in, and her model virus, bacteriophage is SPN3US. This happens to be a very unique bacteriophage. It's called a giant phage. There's more than 50 different proteins in there, which is really unique, and at least 29 are essential for the virion. So SPN3US is also really wonderful to study, because Julie has found ways to manipulate and mutate, for example, to have only the head and without the tail, and at different stages of generation. So she says, this is things we need to find out. What is the copy number? Which of the proteins are essential, and what are their functions? Because again, in bacteriophage, with their wide level of diversity, it turns out that even with a complete genome sequence, if you do BLAST, you may get an agreement homology with nothing. There may be no other entity that matches in any way. So how can you find out what's the function of the protein? Mass spectrometry early on proved to be critically important, partly in helping finding the copy number. And we were among the early, if not the earliest, groups to be using mass spectrometry for bacteriophage work. So here's her hypothesis, that a significant portion of the proteins that make up this giant phage virion are essential, and that they're packaged in the head in a way that they're ejected into the host cell with the phage genome. And that's important for a host takeover. And that's not typical of all phages. So she has something there that is still in the form of a hypothesis, but clearly worthy of following up. And here are her specific aims. I'm not going to read them right now to you, but you can see that she's divided it into a few sub-aims to make it more logical. And I'm not including the methodology she's going to use here, but they are very clearly stated of what she wants to do. And you don't want to just always say to determine if or to identify, but make it a really positive statement. So here's her specific aims. And a really important point here is this order, because specific aim needs to be interesting and important, but clearly doable. If you have something speculative at the beginning, suppose she didn't have a way to really isolate this giant phage. And if you fail on your first aim, then you're dead in the water for the rest. So you need to be sure that that first aim is rock solid and the technology is well established for you. And then you go on and continue of how you're going to solve this hypothesis. And here she's listed her specific aims. When you have then the rest of the proposal, as I had mentioned, there's a significance, innovation, and approach. And in approach is where you have background and preliminary data, and experimental design. Experimental design you have organized according to the specific aims, and you list them and have a rationale, the specific aim, and how you're going to accomplish it. And this is where the wording needs to be exactly the same. So you don't want to have modified one and not the other. Rationale, and I said plan methodology, and this is again where you have pitfalls and alternative strategies. I wanted to show you the examples of Julie's preliminary data from this proposal. It illustrates a few key things. First, it happens to be documenting help by her collaborators. And here, this is the genomics, but it also involved Julie's informatics work. Here is a cryo-EM structure, and it needs to be visually informative even to non-experts. So it's always amazing to me to see a micrograph of a bacteriophage with this tail and head and the often icosahedral head. So it's really quite amazing. And then, of course, I was happy to show some of our mass spectrometry results because there's proteome processing, ProHead protease processing that can easily be determined by mass spectrometry. But what this points out is you need to illustrate what you've done, but the reviewers may not be real experts. They should have knowledge of the field. So you don't want it to be too obscure in terminology that they're not going to understand. And then at the end, after all the other parts, there's anticipated outcomes and future work. I'm obviously not going to read this, but you need to have a way of saying, what are you going to find out? And if it all works, what do you think you'll learn? And then where might you go with it? Because most projects are not going to be just closed-ended, finish funding, and that's it. It would not be that interesting. So fortunately, Julie, at that point, her initial submission had gotten a score of 24, which normally sounds really exciting. At that time, there was tight funding. The program officer wasn't sure if it would get funded or not, and told her to start preparing a resubmission, which she did. But fortunately, she had a paper accepted for publication. And 2 days later, he called and it was funded. So it was funded and we got to do the work. All right, I'm going to tell you just a very little bit about the S10 program, which I'm particularly familiar both as an applicant and a reviewer. We had a webinar sponsored by the Association of Biomolecular Resource Facilities, ABRF, organized by Joe Dragavon, Sheena Mache, and I did it. That was put on in February, and they said that I could use a few of their slides to show you. If you haven't heard of an S10 before, this is a very different type of grant application from with instrumentation. It's the NIH program. And you can see it said it's to support purchase of state-of-the-art commercially available instruments for shared use. Really key point here, commercially available. To be eligible to be purchased on an S10, it has to have a part number. It has to have the manufacturer's typical warranty. It cannot be experimental, beta, prototype. It must have a part. And it needs to be used by researchers predominantly with NIH funding. You can see here that it's a once a year due date. If you haven't started working on one now, it's definitely too late for this year and time to get started for next year's. There's three general categories. First one is called big, but it's really basic instrument grant for $50K to $250. And this is specific for institutions where there hasn't been funding of more than $250,001 in the previous 3 fiscal years. The most common is the S10 program, 50 to 600K, and then the high-end program, 600 to 2 million. Now, you don't have to have something. For example, in an S10, if you choose to put in an S10 and your instrument costs 700, that's okay, but you have to come up with how you're going to pay for the other 100K. There's 3 main justification types for this program. One that's very easy to document. You're replacing an aging instrument that you've used for years. Maybe it's no longer supported by the company. It could even be not working anymore, but you absolutely need a new one. Maybe you're acquiring additional instrument to alleviate capacity or access problems. It's a little trickier to document this, but you can do that. Or you're acquiring an instrument that has new functionality or capabilities in support of research. Now, I submitted an application previous year, and I was really able to use all 3 because I had an old Orbitrap that wasn't going to be supported, and I had doing a lot more data-dependent acquisition MS, and I also got a FAMES source to help with cross-linking MS. I'm not going to go into detail here, but I'd be delighted to help anyone who wants more information about this. You have to justify the need for your users, but also why you picked this specific instrument and what brand. And it has to be the right choice. If you pick one that's more technology than you need or less, it shows that you have not used good judgment and you are not asking for what's best for your users. It's not a case where they can cut the budget. So that will give you a low level of enthusiasm. You have your technical expertise, summarize what the main major and minor users and how they need it. And the buzzword here, the important one, is the impact and how it will advance their research. And then you summarize the usage and any accessories. Very different from standard research proposals. You have to show a lot about the organization and management, where it's going to be, how it's supervised, advisory committee, financial plan for support, data management, sharing. It doesn't need the new sharing plan, but how you manage the data. And institutional commitment is absolutely critical. Cost sharing is not required, so that means there isn't a required amount that the institution has to put into the purchase, but there has to be institutional help in either staff or helping with service contract, and it has to be clear how this instrument is going to be supported and maintained for at least 5 years. Usually the warranty is 1 year, so the 4 after that. And what's noted here is if you have institutional funds available, don't use it for the purchase price, but use it for service contracts. There's an overall benefits section that I usually have as barely a paragraph. And then you have to have letters of institutional support that have dollar number commitments from the institution of how they're going to help support the maintenance. And you have chargeback fees, but you need to really have a clear financial plan. And for example, if it's an instrument that there are many of them either on your campus or nearby that could realistically be used, but they can't, either they're overcommitted or they can't give you access, you need letters documenting that. And I won't go into the subtleties of instrument demos, but it is important as a part of documenting why you've picked what instrument you want. Now, if you have the right circumstances, NIH-funded users need a new instrument, the funding level really is very accessible. And that's just showing about the institutional commitment, reiterating. I've seen many really enthusiastic, outstanding proposals with zero institutional commitment, and the level of enthusiasm of reviewers goes way down, and it's essentially kills them. And then I'm just showing you here, this was my 82 proposal, and I put in a proposal for 22, and it was awarded. I felt really great to have one in the first year of the program and in the 40th year of the program. And I thought it interesting to see the 250,000 from 1982 translates with inflation to almost 780,000, which isn't really all that far off from what I got in 22. Now notice here, there's an extra 90 because the cost went up after the proposal was submitted and the institution had to come up with that. And I say you can do it. It takes a lot of work, but I'm confident that you'll work out how to do it, and just remember to get help from all of your colleagues. This is Texas bluebonnets that are in bloom right now. If we would only get some rain, and I'll be happy to answer any questions.
Thanks, Sue. That was an exciting, fantastic talk. I would like to open up to the audience to put in questions into the chat. And we can start off, Giuseppe and I can start off a little bit early with a few questions. Giuseppe, do you want to go first or should I?
I hope I'm not getting graded.
I can go first. Well, first of all, I would like to thank Sue for being here today and talk to us about projects and so on. So I have a few questions actually. I'll start with the first one, and it's about what do you think about including the limitation of the study or the type of projects that you are doing? Let's say you are having few steps in your either experimental design or so on. What do you think about putting also limitation in a research project?
Well, I think that may go into the pitfalls section in a way. You have to be realistic. And so the key is to convince the reviewers that you know what you're talking about and that your expected data can be obtained by the methods that you're using. You know, for example, we do a lot of discovery proteomics with DIA-MS. By our standard techniques, we're not going to get unexpected post-translational modifications. And if that's something that I need, you definitely don't want to have that in there. And you would say, this is not a method that we're going to use for PTMs.
Yes, because in your presentation, you talk about actually being innovative, but until a certain point, not actually putting something that has to be over. And so something that can be seen as a potential project to be funded. So yes. I think
there is what they call high risk, high gain aspect. You can have a somewhat more risky, for example, specific aim 3 that is building on your other aims that will lead. And also you put that in your future studies of where you're going to take the results from this and move it forward. Because you really don't want it to be, as they say, over ambitious. So you don't want to promise more than you realistically can do. And I didn't even go into the aspects of statistics and sample sizes. You really need to document if you're doing anything with human subjects, for example, why the numbers that you're picking. Make sure you have consultants in those areas because we can't be expected to know it all ourselves.
Thank you. I don't know if there are some questions from the audience. Can't see here.
I'll give one just
Yes,
while
please.
we're waiting. So you mentioned interdependence and aims as being a really important point. What's the best way to deal with aim interdependence when your first aim is discovery proteomics?
Well, that's a tough one. I don't,
because most of our studies at least have some component of discovery proteomics, right?
There needs to be something more substantive than just starting with discovery proteomics, because in your experimental set, you may not find, for example, pathways that are overrepresented. You may find some individual proteins that are changed. And in that case, I really feel you would need to have preliminary data that has given you an idea that additional discovery proteomics is going to flesh out what the differences are you've seen in your pilot studies.
Excellent. Yeah, I think that that's a really important point, that we need to preliminary data with regards to discovery proteomics is really necessary for those types of proposals, right?
And many times people use the discovery proteomics in a cyclic way where they start and that gives them a lead. Then they have a biological study, then a manipulation, then they go back to the discovery proteomics to see how have those manipulations impacted the system that they're investigating.
Along those lines, do you think can you give like what are the most common mistakes that you've seen in grant applications you've reviewed that might be pertain to proteomics people or to mass spec people?
Unrealistic expectations for the technologies that they're going to use, incorrect choice of methodology. All right, I will be really extreme in this, but let's say someone has a triple quad in their lab and they've been using it for targeted analysis, and they say, well, we're going to go do discovery proteomics with this, and reviewer who has any experience is going to say, would be worth it to do discovery proteomics, but you're not going to do it the way you did it. I think unrealistic expectations for whatever technology that you're picking. So it's a balance. Pick the right technology and have realistic expectation. But again, something that I didn't emphasize, it's so important to have a colleague read your proposal before it's finished. Make sure it's clear and make sure it comes out, what are you trying to find? Because it is obvious to you what your overarching question is, but it may not be for others. If English is not your first language, accept the fact that someone is going to tear it apart and give you lots of editorial changes, because it has to be understandable. People understand, they recognize that not everyone is going to be writing perfect prose, but it can't have tons of mistakes. I didn't mention spell checker. Absolutely, make sure you don't have typos and spelling errors. It's inexcusable these days with word processors.
I think we have two questions from the audience, so we'll start with the first one. Which section of the grant do you suggest we should start writing first? And what do you think will be then the order to write after that?
Well, I first outline the hypothesis in question, but the hypothesis will keep evolving as you work out the methodology. Then I like to say what methods are being used. Then you go back to the specific aims. I have these methods, I have this question. how will I formulate and organize these into convincing aims for the project? I wait to write the introduction and background until I'm absolutely sure what it is that's going to be studied. And then I don't do the abstract until the very end to make sure what's going to be covered. So it's a very iterative process of having it all work together, because it has to flow and it has to make the point and be convincing. Because remember, many of the reviewers will have 10 or 12 grant applications that they're assigned to. So you need to be sure that the text is clear and that they can find something to say positive about it, of what you want to do.
On the same line, actually, in your presentation, you pointed on the importance of the abstract, which is something that really should capture the attention of the reviewer. And on the same line, there is a question from Justina, which is also part of the ETC. She thanks you for the presentation, and she's asking, what do you think about including a summary figure in the aims, like a graphical abstract?
It's not usually done. There's not very much space. It's a small area, very specific. I personally think words, it's not a case of a picture is worth 1,000 words here. I really think you need the words.
Yeah, so I guess I've got a couple other questions. So with NIH proposals, particularly R01s, there are specific requests for applications, RFAs, and then there are sort of more generalized ones. How much tailoring do you need to do to a specific RFA? And then what is the trade-off between a specific topic RFA or a more general submission?
Well, when it's a specific topic, of course, you're going to be competing against people in a very aligned area, but you're also more likely to have experts in the review panel who will know that area. These are things where I feel contacting the program officer is the best, because they're going to be able to guide you. Is your topic really suitable? Now, there's lots of resources on NIH. I didn't have time to actually show you the sites, but you can find the list of institutes. Then you click on the institute, they have their current funding opportunities. For that, you can find the program officers, and they really will be delighted to work with you. So I think they're the best ones to guide there. There are certain study sections and general plans. And this is where talking to your colleagues, your senior colleagues, they may know, for example, oh, if it goes to such and such a study section, they just don't like to fund, let's just say, proteomics work. I mean, they're all clinical. So you need to use all the resources that you possibly can to help guide you. But plan, plan, plan. Don't start writing until you really have everything else worked out.
There are actually another two questions from the audience. One is regarding how can we present possible findings of basic research as an interesting output when they are not related to translational research?
Well, basic research, if it is, first off, you have to find the right funding agency, because NIH may not be the right place, but for example, NSF may be ideal, because there you really don't want to have the biomedical application. In fact, a colleague of mine here talked about a proposal that he submitted and was successful with NSF, and he essentially took out every word of the disease he was interested in, because he also had very good basic biology. So you have to pick the right funding agency. And there's also small foundations. This is where if you have an active grants management sponsored program office, they can often help you find something to support your research.
Actually, it's very interesting because this question goes along with what you said in the beginning, just to read exactly the funding, the call, and actually tailor the application towards specific objective of that call. I think
Yes.
This should be, and not only in US, but also in several countries, as you said, there are specific funding agencies for basic, what we call it basic research, or more translational one, or medical research.
Because, I mean, we all know we still need the basic research, and it can't always be translational. You don't want the translational aspect to be superficial and just stuck on there to make it interesting. So it's just finding the right fit.
Thanks. There is, I will take another question from the audience, Brian, if it's okay.
Please.
In case you have a gap in your publication record, like due to unforeseen circumstances, can we put that in the letter when we are applying for a grant?
I would say the best place to put that is in your personal statement in the BioSketch. If this is NIH, I don't know for others, but you have that place for a personal statement, and that's just where you can say anything that you want the reviewers to read. And especially if you're the PI, that will be read. So it's something very important. That's something you also want to have your colleagues look at and make sure it's okay.
And also following on that question, based on your experience, do you think that like an interruption or a gap in publication can have an effect in the scoring of the application?
Well, it shouldn't. If you have a gap and have come back and are republishing, and there's whatever the reason for the gap, that they should not take that into consideration. It just needs to be clear and open. Remember, the reviewers are us. We all have been in that same place, and life happens. So it has to just be a reasonable explanation. And I'll tell you kind of in a story, a dear colleague of mine who is even older than I am, had submitted an application, and he had turned 80. And very inappropriately, one of the reviewers mentioned something about retirement, that he thought he was retired. And so he resubmitted and had a letter from the president of the institution documenting that he is still active and here every day and working, and he got funded. And he had an R01 funded after 80. And it's absolutely amazing. So
that's an incredible story. That's a great note, actually, for us to close on, since there aren't any other questions and we've hit the hour. So,
but
Sue,
I will be happy to anyone who wants to contact me directly, particularly about S10s, which is something that I do have much detailed experience, but reviewing and supporting other people's R01s from mass spectrometry. So I'd be delighted to help anytime.
Excellent. Yeah, so for everyone else, she's Weintraub at UT hscsa.edu.
Can you put it in the chat?
Yes, I will put it in the chat here to everyone. And that she's early enough to get into email so that she has her full last name as the, that's it. That's no numbers, no abbreviations, no letters.
Well, there's not many Weintraubs in here, unlike the East Coast.
But excellent, Sue. This was a fantastic session. I really appreciate you taking the time to do this. Hopefully everyone else here on this session will write better grants as a result.
I certainly hope so, and I'm happy to help anytime.